Hi everyone, so today we are going to look at CRISPR-Cas9. So this is a little bit more of a complex process, so take your time with this, slow down parts if you need to, rewatch parts, and as usual, bring your questions to class. So the key knowledge from the study design is the function of CRISPR-Cas9 in bacteria and the application of this function in editing an organism's genome. So why is this important? Well, CRISPR-Cas9 is naturally found in bacteria, and we've been able to use this process to manipulate DNA very precisely. And this is one of the ways we create GMOs. And so we're looking at all the different ways that we can manipulate DNA. So let's find out how CRISPR-Cas9 works and how we use it to manipulate DNA. So what is CRISPR-Cas9? Well, it is naturally a bacterial defense system. So CRISPR stands for clustered, regularly interspaced short palindromic repeats, which sounds like a lot, but let's break it down. First, let's look at this image over here. So here we have a CRISPR array, this part here. It's a sequence of DNA found in bacteria that is used for immune defense. So it is clustered together. It is regularly spaced out. So we've got a repeat, then a spacer, a repeat, then a spacer. It's regularly interspaced, and it has short palindromic repeats. So over here we have the repeats. They are short and they are palindromic, meaning that they are read the same from the forward here, and then they're read the same backwards. So a palindrome is something that is read the same forward and backwards. For example, the word race car is read the same forward and read the same backwards. Same with the name Anna. You can read it as Anna forwards or Anna backwards, right? So that's a palindrome. We've got clustered DNA sequences that are regularly interspaced, and they have short palindromic repeats. And let's get rid of all of this. Okay. So CRISPR, as I said, is naturally occurring sequence of DNA found in bacteria, and it plays an important role in the defense against viral attacks from bacteriophages. So a bacteriophage is a virus that infects a bacteria. And so CRISPR is involved in defending the bacteria against these viruses. So when a bacterium encounters this virus, it takes like a mugshot of it, so kind of like a photograph of it, by storing some of its viral genetic material within the bacterium's own genome. So in these spaces, the bacteria will take part of the virus's DNA and put it into this spacer to help it remember that virus, and that is then used in its immune defense. So the next time that virus invades that bacteria, it's able to transcribe this mugshot in the spacer region of CRISPR, and it attaches it to an enzyme, an endonuclease enzyme called Cas9 over here. And this enzyme here is involved with destroying the invading virus. So how does the CRISPR-Cas9 work? So we looked at the CRISPR complex or the CRISPR array, and we looked at the endonuclease that is Cas9. So they work together. But how does it work together to help the bacteria fight off a virus such as a bacteriophage? So this is a bacteriophage virus that infects bacteria. So first we have this bacteriophage virus injecting its DNA into the bacteria. So this strand here is the viral DNA. We have enzymes, so endonuclease enzymes called Cas1 and Cas2, that look for PAM sequences in the viral DNA. So what is a PAM sequence? Well, a PAM sequence stands for protospacer adjacent motif. And so right next to this particular section, we have two PAM sites or PAM sequences. And a PAM sequence is a sequence of nucleotides next to the target viral DNA, which is recognized by the bacteria's immune defense. Okay, so like we said, in the CRISPR array, we have spacers. So proto means early, so a protospacer is an early spacer. So we said that the bacteria will take a mugshot of the viral DNA and store it in the spacer in its CRISPR array. So the protospacer is that sequence of viral DNA that it is going to store in the spacer region. We call it a protospacer because it's not yet stored in that spacer region, so we can't call it a spacer yet. So we call it a protospacer, meaning early spacer. And the way that Cas1 and Cas2 find this protospacer, this sequence of DNA that we're going to store, or the bacteria is going to store, is by looking for a PAM sequence, so a sequence of nucleotides on either side of this protospacer. That's why we call it a protospacer adjacent motif. So adjacent meaning next to, so series of nucleotides next to the protospacer that Cas1 and Cas2 recognize. Then Cas1 and Cas2 will make blunt end cuts at the PAM site of the protospacer. So at these PAM sites, we get blunt end cuts by Cas1 and Cas2. And now we have this specific sequence of viral DNA, which we call the protospacer. Cas1 and Cas2 then insert this protospacer into the 3' end of the CRISPR array. So remember that the CRISPR array is a sequence of DNA in the bacteria. And in this sequence of DNA, there are spaces, and we insert the viral DNA into this space, and we call it a spacer. And this is what the bacteria uses to defend itself against the virus. Once the bacteria has inserted that spacer, RNA polymerase will then come along and it runs along the spacer and creates a RNA molecule, which is then modified and becomes a single guide RNA. So we can abbreviate that to sgRNA. And so that should be SG. So the sgRNA contains a part of the palindromic repeat, which then creates a hair loop or hairpin loop like this. And this section then helps attach or anchor to Cas9. So it sounds like a lot, but let's briefly run over it again. So bacteriophage, this virus, injects its DNA into the bacteria. On this viral DNA, we have PAM sites, which are a sequence of nucleotides that are next to the protospacer. And we want to take this protospacer, so Cas1 and Cas2 will find the PAM sites to then cut at those PAM sites. So we take this protospacer, or the bacteria takes this protospacer, inserts it into the CRISPR array as a new spacer. RNA polymerase comes along and transcribes that spacer to create a single guide RNA. Part of the single guide RNA is palindromic, so it is read the same forwards and backwards, which means it's complementary to each other. So they pair up together to form this loop. And this loop is important because this helps anchor this guide RNA or single guide RNA onto Cas9, which then is used in immune defense. So let's keep going. So we have the single guide RNA. We have the single guide RNA with this hairpin loop, and it joins to an endonuclease, which we call Cas9. When they join together, they form a Cas9 complex. And Cas9 will use this guide RNA to compare to DNA it encounters to find complementary viral DNA. So it's going to go out and look for any viral DNA that may exist in the bacteria that matches or is complementary to this guide RNA. Because when the bacteria is infected with that specific virus again, this Cas9 complex will recognize it using that specific PAM sequence. So remember, the Cas1 and Cas2 will look for a PAM sequence. Cas9 will also look for that specific PAM sequence on that viral that viral DNA. And then this complex, Cas9 complex, will compare the guide RNA to that virus's DNA to see if it is complementary. So it's going to look for these PAM sequences. Cas9, then, once it's found those PAM sequences, it's compared the guide RNA and it goes, yes, this is the viral DNA that I've been looking for. It will create a blunt end cut. And so it will cleave the phosphate sugar backbone of the DNA right here. And this inactivates the virus and prevents infection, because we know that DNA is the code for proteins. So if the virus cannot make its proteins, it cannot be used. So it's useless. Cas9 contains 2 active sites. So it will cut both strands of the DNA and create that blunt end cut. When the viral DNA is cut, ligase, as we've learnt about, will naturally try to repair it. So it will naturally try to repair like this. However, we hope that during this process, ligase will make an error, and this will cause a mutation in the sequence of DNA, and therefore the viral genes are non-functional, so they won't work. So we're hoping that this natural process of the DNA being cut and then ligase repairing that DNA will be an error. So an error will occur. If this doesn't happen and ligase just repairs the DNA normally and it's all perfectly fine, and it repairs that viral DNA to be normal, what will happen is that this Cas9 complex will repeat this sequence, or repeat this process. It will look for the PAM sequence, it will compare the guide RNA, it will make that blunt end cut once it realizes it's found that viral DNA, make the blunt end cut, ligase will try to repair it. We hope that a mistake is made while ligase is repairing it. And if a mistake is made, then the viral DNA is useless, it is non-functional, and therefore the bacteria has successfully defended itself against a virus. So how do we use the CRISPR-Cas9 system? Now we know that is a natural bacterial immune defense. Why is that important to us? How can we use it? Well, scientists have been able to adapt the CRISPR-Cas9 system to edit genomes with great precision. So the CRISPR-Cas9 system can induce genetic changes by cutting DNA at a location specifically chosen by scientists. So we know that CRISPR-Cas9 will cut DNA at a specific PAM sequence. And so we can make synthetic guide RNA to guide Cas9 to that specific section we want to cut. And this creates an opportunity for nucleotides to be added, removed, or substituted into the selected sequence that we want to cut. In turn, this can then knock out, enhance, or change the function of a gene. So if we want to change a gene, we can create the single guide RNA, we can do it synthetically, we can combine it with Cas9, and we can insert it into a cell. And then that Cas9 complex is then going to go look for a DNA sequence that matches that guide RNA. And once it finds it, it's going to cut at that specific spot. Then we can add in or remove or change whatever sequence we want at that single spot. So if we want to add in a sequence that will benefit a corn plant, for example, we can do that using the CRISPR-Cas9 system. Currently, this is the most precise and the cheapest option for gene editing. So these are some of the ways that we currently or can use CRISPR-Cas9. We can use it for research. So for example, attaching a fluorescent protein to Cas9 to locate a specific gene in the genome. So we can use it to figure out where a specific gene is because your genome is so huge, or an organism genome might be really huge. You can use it for disrupting the expression of a gene to see the effect that that protein being knocked out. This helps identify the function of specific genes if we're able to knock out or silence a specific gene. We can also use it for dealing with disease, so replacing a deleterious allele with a healthy allele, adding genes that code for proteins that decrease susceptibility to infectious disease such as HIV or AIDS, modifying cancer-promoting genes to make them less influential. We can also use it in agriculture, which we're going to go into a little bit more detail with. So introducing pest and herbicide-resistant genes to increase the yield of crops, and altering genes to promote increased growth to improve the yield of crops. So this seems like a lot of different uses. I want you to know that we can use CRISPR-Cas9 to edit genes for research, for dealing with diseases, and agriculture. So I wanted to compare how CRISPR is used naturally occurring in bacteria and how CRISPR can be utilized in a lab. So let's start with bacteria first. So we know that viral DNA is injected into the bacteria. This is incorporated into the spacer region of the bacterial DNA. This DNA sequence is transcribed into guide RNA along with the special PAM sequences. These PAM sequences are recognized and attached to Cas9, which is an endonuclease that will cut the DNA in specific spots. This then allows Cas9 to recognize foreign viruses and cut their DNA. We know all of this. And then when DNA is cut, it automatically tries to repair via ligases, not very good at doing their job. So we know ligases can make mistakes, and it's hoped that there are mutations that will occur during this process to render the viral genes non-functional. So the bacteria hopes that ligase will make a mistake. This is a diagram that kind of sums that up. You can pause the video now to look at that in more detail, but this is what we've gone over so far. How bacteria use CRISPR-Cas9 in its immune defense. So we've talked about how the uses of CRISPR to us and how it can be used in a lab. This is how we would go through that process. So we start with a DNA sequence for a gene of interest. Example, we might want to be looking at a cancer gene, and we want to isolate it and replicate it by, we isolate and replicate it in a petri dish, so in a lab essentially. This guide RNA is produced, so synthetic guide RNA is produced, which is then mixed with the Cas9 enzymes, and they bind together to form a complex. So we make the guide RNA, mix it with Cas9, we form a complex. The guide RNA identifies the specific DNA sequence of interest, and it leads Cas9 to cut that sequence, the same way it does in a bacterium. Ligases will then automatically try to repair the DNA, and this is where scientists can either replace the faulty gene with a new DNA or new gene. So if we're looking for this cancer gene, so we make a guide RNA that is complementary to this cancer gene, and we mix it with Cas9. So this Cas9 complex is looking for this gene. Once it gets to that gene or that sequence of DNA and cuts it, we can either replace that gene altogether with something new, or the ligase attempts to repair the gene, causing a mutation in the DNA, silencing the target gene. So we're kind of hoping for ligase to make a mistake and silence that target gene, so that cancer gene no longer is functional. Again, this is a diagram that sums it up. So we get the guide RNA, we create it in the lab. We mix it with Cas9, which is also known as molecular scissors, which I kind of like because it is cutting DNA. Cas9 is added to the guide RNA mix, we make that complex, and it's injected with guide RNA, and the Cas9 mix, that mix is injected into cells. Guide RNA identifies the mutated gene, Cas9 cuts out the mutated sequence, there's a double-stranded break in the target DNA, as you can see down here. Then we can either have a faulty gene be replaced by a corrected gene or a new gene altogether, or we can allow the cells to attempt to repair it and hope that a mutation occurs, silencing it. So I want you to know how it naturally occurs in bacteria, and then how we can use that process, which is essentially the same thing, to modify genes in organisms in a lab. There are some limitations of CRISPR. So we've talked a lot about how it can be used, and it can be used in a multitude of different ways that are super beneficial to us and to research and to agriculture. So what are the limitations? We've mostly only had animal studies, so no human studies yet. Human studies are very complex. Editing genes in humans, there's a lot of controversy surrounding it, so we haven't had human studies yet. CRISPR cuts DNA at a chosen site, but scientists still have to introduce nucleotide sequences of interest and hope that it gets incorporated. So we can't be sure that it is. We just hope that it is incorporated. So we know that it will cut at that specific site, but when we're introducing a new nucleotide or DNA sequence, we hope that it gets incorporated. And to successfully alter an organism's genome using the CRISPR-Cas9 technology, scientists must treat an embryo prior to the cells differentiating. So think back to when we looked at stem cells and how stem cells can become any type of cell. We have to inject those cells at a very early stage of an organism's life as an embryo. We have to inject the CRISPR-Cas9 complex before it starts to differentiate. And this will then ensure every cell in the organism is altered. So it has to be done very, very, very early. There are many ethical considerations, as you can imagine, with using CRISPR, and it's quite controversial. And if you read news articles about it, you will find that there is a lot of ethical discussion about using CRISPR. So firstly, safety. So there is a possibility of off-target cleavages, so edits in the wrong place or mosaics, so some cells containing edited genomes and others not. And this means that many scientists are hesitant to use CRISPR outside of research. All right, so it's really only used for research right now. Informed consent is also another ethical consideration. So scientists cannot get consent from embryos to edit their genes. So if the embryo goes out to be born and one day has children of its own, these children will also never have consented to scientists interfering with their genome, because obviously embryo cannot consent. How can we make edits to an embryo's gene? If we're talking about humans here, how can we make edits to a human embryo genome before that person is even alive? So we can't get that consent, and consent is very, very important when you're doing things like this. Inequality, so there is concern that only wealthy people will be able to afford the use of CRISPR to treat genetic conditions or otherwise change their genes. So this technology may be only available to certain groups, mostly wealthy people. And also discrimination. So CRISPR may be a threat to those who are judged by society as biologically inferior, when in fact these individuals do not feel that they need fixing at all. So there might be people who want to make sure that the embryo doesn't have a specific gene or a specific trait, and they edit their baby's genome, so using CRISPR, so it doesn't have that specific trait. But obviously groups in society might think, actually, this is not a bad thing. We shouldn't be editing people's genomes, or they might be opposed to it because it's saying that anyone with this trait might be biologically inferior. For example, some people inherit genes that cause them to not have any hearing, so that they are deaf. So if a group or parents decided that they wanted to make sure that their child was not deaf, and they edited their embryo genome so that the embryo would become a child that has full hearing, well, the deaf community might be opposed to this, because it's like those parents or those groups are saying that being deaf is biologically inferior and they need to be fixed, so we shouldn't have any deaf people, which is not true at all. So as you can see, there are multiple different points of views that need to be considered, many ethical considerations that need to be considered when using CRISPR, because it is a very new technology and and has a lot of controversy around it. Alrighty, so as usual, write down your questions. I know this is quite complex, so rewatch parts if you need to, slow things down, make your notes, and bring those questions to class. And I'm going to leave you with this funny meme from Amoeba Sisters, who I love, as you know. And I will see you next time.